friends, today we will be discussing the process of glycolysis, the products, the complexes formed during the process of glycolysis, the enzymes used, the names of the process and the enzymes involved, and the net ATP or the net energy released. So, the first thing that we will be discussing is the ATP formed or the ATP consumed and in which steps they are done. So, the first molecule of ATP is consumed in step first. The step when glucose is converted into glucose 6 phosphate. In this process, one molecule of ATP is consumed. The next when ATP is consumed is the process when fructose 1,6 phosphate is converted into fructose 1,6 diphosphate. In this process, one molecule of ATP is consumed. And in these process, the conversion of glucose to glucose 6 phosphate and in fructose 6 phosphate to fructose 1,6 diphosphate, you must be noticing that one molecule of phosphate is added. And when phosphate is added, the name of the process is phosphorylation. And here also it is phosphorylation. Whenever the phosphate is added and the process of phosphorylation takes place, one molecule of ATP would be consumed. Now we would be seeing where the ATP are formed. So the very first process where ATP is formed in this, when 1,3-diphosphoglyceric acid is converted into 3-phosphoglyceric acid, that is, one phosphate is removed. When the phosphate is removed, then the ATP is formed. Now I will be writing 2 ATP. Now you must be wondering why I have written 2 ATP. So you must be seeing that this fructose 1,6 diphosphate has been splitted into glycerol 3 phosphate and dihydroxyacetone phosphate. These both are isomers of each other. So these two molecules would be doing the same thing. So one ATP would be formed from glycerol 3 phosphate and the other molecule would be formed from dihydroxyacetone phosphate. So I have written here 2 ATPs are released. Now the another set of ATP are released in the process when 2-phosphoenol pyruvate changes into pyruvic acid. Here also we would be writing 2 ATPs are formed. How much? 2 ATPs. Now the ATP is consumed and the ATPs formed are discussed. Now we will be discussing where the another unit of energies are formed, and I am talking about NADH2. So when this glyceraldehyde 3 phosphate converts into 1,3-diphosphoglyceric acid, then 2 molecules of NADH2 are formed. NADH2 are also the forms of energy just like ATP. It's just that NADH2 are bigger forms of energy. Now we would be writing the names of the enzymes that are important. That would be very important for your Neet exams. So the very first step in which glucose is converted into glucose 6 phosphate, that is in the phosphorylation step where one ATP is formed. The enzyme used is hexokinase. Which? Hexokinase. Wait, let me use another color for this. Otherwise you would get confused. So yeah. Which enzyme is used in the very first step? Hexokinase. Now in the second step, as you are noticing that glucose 6 phosphate is converted into fructose 6 phosphate. That is, in this process only isomerization is taking place. So the enzyme would be isomerase. Which enzyme? Isomerase. Now in the next step, when again phosphate is adding, that is the step of phosphorylation, the enzyme would be kinase. Remember that Whenever phosphorylation takes place, kinase is used. Now, in the next step, when the splitting of fructose 1,6 diphosphate takes place into glyceroldehyde 3 phosphate and dihydroxyacetone phosphate, the enzyme used is aldolase. This enzyme is really important, remember this. Now, in the next step, when NADH2 is formed, let me tell you more about this step. This process is dehydrogenation. Dehydrogenation requires the enzyme called dehydrogenase. In this, removal of H2 takes place. Removal of H2 takes place and addition of inorganic phosphate takes place. This step is also very important, and this is an oxidation process. Now, in the next step, when 1,3-diphosphoglyceric acid is converted into 3-phosphoglyceric acid, and you must be noticing that one phosphate has been removed, and whenever the removal or the addition of phosphate takes place, the enzyme is kinase. Which enzyme? Kinase. Now, in the next step, when 3-phosphoglyceric acid is converted into 2-phosphoglyceric acid, and you must be seeing that these are the position isomers of each other. So, the enzyme would be mutase. Which enzyme? Mutase. And now, in the next step, when 2-phosphoglyceric acid is converted into 2-phosphoenol pyruvate, enol form is formed. So, the enzyme would be enolase. And in the very last step, when 2-phosphoenol pyruvate is converted into pyruvic acid, you must be noticing that phosphate is again removed. And whenever phosphate is removed or added, the enzyme used would be kinase. Which enzyme? Kinase. Now, as we have discussed the steps and the energy released or consumed and the enzymes, now we would be, as a sum up, I'll tell you first what we have done till now. 2 molecules of ATP, one molecule here and one molecule of here, are consumed, and then 2 molecules of ATP, and then these 2 molecules of ATP are released, and this two molecules of NADH2 are also released. So I'll be writing here the consumed things and here the released things. Consumed are 1 ATP and then 1 ATP, yeah, and the released one would be 2 ATPs, 2 ATPs, and then 2 NADH2. NADH2. Okay. Now I will be changing the slide and we would in detail discuss this, the net energy released or the net energy consumed. So as I've told you that what all are consumed, 1 molecule of ATP and then this molecule of ATP, and what are released, these 4 molecules of ATPs and 2 molecules of NADH2. Now you must be familiar with the ATP forms, but what is this NADH2? How many ATPs are equivalent to this NADH2? And we would be knowing this with the help of the shuttle system. And with the shuttle system, I mean these NADH2 are converted into ATPs via EDS. EDS is electron transfer system. We will be discussing this system also in the later videos. But now we will be discussing how many NADH2 are equivalent to how many ATPs. So this depends on the shuttle system. This depends on the shuttle system. Now there are 2 types of shuttle system. I have changed the pen so it would be more clear. Shuttle system are of 2 types. The first is the mallet one, and the other one is the glycerol phosphate one. Which one? Glycerol phosphate. Now, in the mallet shuttle, 1 NADH2 is equivalent to 3 ATPs, and in the glycerol phosphate shuttle, 1 NADH2 is equivalent to 2 ATP. Now you must be wondering why this is happening. So in the mallet shuttle, NADH2 remains in the form of NADH2, but in glycerol phosphate shuttle, NADH2 converts into FADH2. And this FADH2 is equivalent to 2 ATP, hence you will be gaining 2 ATPs in the glycerol phosphate shuttle. Now, this mallet shuttle is found in liver cells, liver, heart, and kidney. And this glycerol phosphate shuttle is found in the nerve cells and the muscle cells. These are really important as in previous NEET exams, these questions have been asked very frequently. Now I'd be changing the slide and we would be discussing this again. So what we have discussed earlier is, first we would see released energy. Total 4 ATPs I can write, right? 2 ATPs from the first step and the 2 ATPs from the last step. So 4 ATPs and then 2 NADH2. 2 NADH2 would be equivalent to either 6 ATPs or 4 ATP, as I told you about the shuttle system. See, and 3 ATPs I'm talking about when mallet shuttle is present. mallet shuttle में एक NADH2 3 ATP के बराबर होता है, तो 2 NADH2 कितने ATP के बराबर हो गया? 6. और ग्लिसरॉल फॉस्फेट शटल में एक NADH2 2 ATP के बराबर होता है, तो फिर 2 NADH2 4 ATP के बराबर हो गया. दिस इज बेसिक यूनिटरी मेथड. So adding them, either we would be getting 4 plus 6, that is either 10 ATP or 4 plus 4, 8 ATP, depending upon the shuttle system. Now we're adding the consumed things. Consumed is very easy, only 2 ATPs are consumed, ATPs are consumed, right? So the net would be 10 ATP. First we're talking about the mallet shuttle. 10 ATPs are released, and then how many are consumed? 2 minus 2. 10 ATPs are released and 2 ATPs are consumed, so the net would be 8 ATPs. And when the shuttle is the glycerol phosphate one, then 8 ATPs are released. How many? 8 ATPs are released and 2 ATPs are consumed. Then 6 ATPs are net released. So whether the shuttle is mallet or the glycerol, ATPs are always released. In mallet shuttle, 8 ATPs are released, and in glycerol phosphate shuttle, 6 ATPs are released. So to sum up, we will be going back to the previous slides. We have discussed what glycolysis is, right? We have not actually discussed what glycolysis is. I'll tell you in very short. Glycolysis is a breakdown of glucose into pyruvic acid. As you can see, glucose into pyruvic acid, right? In this process, glucose is partially oxidized into pyruvic acid, and in the process of glycolysis, no oxygen is required, yeah? And this process occurs in cytosol, or you can call it as cytoplasm of the cell, yeah? Cytoplasm of the cell. Glycolysis is a chain of 10 reactions. I will be quickly telling you. In the first step, glucose is converted into glucose 6 phosphate, and as phosphate is added, so the process is called phosphorylation, and ATP is consumed, and the enzyme that is used is hexokinase. In the second step, glucose 6 phosphate is converted into fructose 6 phosphate. This is an isomerization process, so the enzyme would be isomerase. In the next step, fructose 6 phosphate is converted into fructose 1,6 diphosphate. This is also a process of phosphorylation where the phosphate is added, so the enzyme would be kinase and 1 ATP would be used. In the next step, fructose 1,6-diphosphate splits up into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, right, with the help of aldolase enzymes. These two are isomers of each other. In the very next step, 2-NADH2 would be released and the process would be dehydrogenation, removal of hydrogen. So glyceraldehyde 3-phosphate converts into 1,3-diphosphoglyceric acid. And a very important thing, in exams they do not give you this glyceraldehyde 3-phosphate, they give you PGAL. What they give you? PGAL. And PGAL means 3-phosphoglyceraldehyde. It is same as glyceraldehyde 3-phosphate, but in exams they use PGAL as short forms. Okay, so 2-NADH2 are used and 2-NADH2 are released in the process, and the enzyme would be dehydrogenase. But then 1,3-diphosphoglyceric acid converts into 3-phosphoglyceric acid, and you can see as ATP, as the phosphate is now removed, so this is also the process in which kinase enzyme would use. As I've told you, kinase would be required whenever phosphate is added or removed. So 2 ATPs are released and then 3-phosphoglyceric acid is converted into 2-phosphoglyceric acid. These are both the position isomers of each other and the enzyme would be mutase. Then 2-phosphoglyceric acid is converted into 2-phosphoenol pyruvate. Enol form is formed, so the enzyme would be enolase. And then the very last step, when the 2-phosphoenol pyruvate converts into pyruvic acid, and the enzyme would be kinase as phosphates are removed, and so the number of ATPs that are released are 2. So if we quickly revise this, so now we would be discussing the gain and the release of the ATPs or the net energies. So the net energy released are 4 ATP. We are not talking about that, we are talking about how much ATPs are released. 4 ATPs, 2 in the last third step and the 2 in the last step, right? So total 4 ATPs and then 2 NADH2. So 2 NADH2 की कहानी मैंने आपको बताई ही थी. This depends upon the shuttle system. This depends upon the shuttle system, whether the shuttle is mallet or the crystal phosphate one. In the mallet one, 1 ATP, 1 NADH2 is equivalent to 3 ATP, and in the glycerol phosphate shuttle, 1 NADH2 is equivalent to 2 ATPs. So the net release either would be 10 ATPs or the 8 ATPs depending upon the shuttle system. Now consumed ATPs are fixed. 2 ATPs would be consumed no matter what shuttle ride. So the net, this is the important one, net gain of ATP. This is very important, you have to remember this, net gain of ATP. So 10 ATPs in the mallet shuttle would be released and 2 would be consumed, so the net would be 8 ATPs. 8 ATP, these are the released one, don't get confused, release ATPs. 8 ATPs are released in the mallet shuttle, and in the glycerol phosphate shuttle 8 ATPs are released and 2 ATPs are consumed, so the net would be 6 ATP. So this is the end of the glycolysis process. Next we would be going for the Krebs cycle. So thank you for today. Revise this. This is really important for Neet.